Hello, everyone, and welcome to today's webinar titled Functional Consequences of the Human Enteric Virome in IBD. This webinar is part of the 2022 Microbiology Virtual Week, and I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during your presentation. You can find that live chat located at the right of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. Now, if you have any trouble seeing or hearing this presentation, just click on that Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located in the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Kate Jeffrey, the Executive Director of Moderna Therapeutics. For a full biography of our speaker, please visit the Biography tab at the left of your screen. Dr. Jeffrey, welcome. You may now begin your presentation.
Hi, everybody. It's a pleasure to introduce to you today our work on the intestinal virome, where we ask questions about what viruses in the gut do, both in health as well as in inflammatory bowel disease. So, viruses have all been on front and center of our minds in the last couple of years, one virus in particular, but it's worth reminding this audience that, viruses are really the most abundant biological entity on Earth. There is estimated to be 10 to the 31 viruses. That's a lot of zeros. And to put that into perspective, that's estimated to be more than the number of stars in the sky or grains of sand on Earth. So there are a lot of viruses. I think our challenge in this space is really annotating a lot of these viruses. There's very few viruses actually annotated. If you look in the NIH virus database, I think it's of the order of over 5,000. So we really are lagging behind in the identity of many of these viruses. But our questions sort of arose by really being intrigued by the fact that us humans harbour many viruses when we're asymptomatic. And sort of akin to the microbiome, we sort of asked whether these viruses that happily exist amongst us with seemingly causing no pathology, we wanted to know whether they educate our immune system in sort of beneficial or maladaptive ways. So it's fair to say within commensal microorganisms, the last 20 years of research has focused almost exclusively on the immunomodulatory capacity of bacteria that inhabit the intestine. But it is worth noting that all of us in this audience probably harbour anywhere from 1 million to 100 million viral-like particles per milligram of our feces or intestinal tissue. So we're harbouring plenty of viruses. Many of those are obviously prokaryotic viruses that inhabit the bacteria, but some of those are actually eukaryotic viruses that inhabit our human cells, seemingly without causing any damage. And many of those therefore challenge Koch's postulate. So they are not only found in people with disease, they do not transfer disease. Some of these viruses just in more than 90% of the human population. So if you take a virus such as virus, which is a single-stranded DNA virus, many of us harbour this without much manifestations of this particular virus. But the presence of these viruses in the gut, it's becoming clear that they actually do have a protective function. So in mice, at least, if you eliminate the viruses in the gut of a specific pathogen-free mouse, or you actually knock out known virus receptors that would sense those viruses, such as the toll-like receptors or cytosolic sensors such as MDA5 or RIG-I, those mice actually have exacerbated intestinal inflammation. So getting rid of those viruses or the ability to sense those viruses is actually damaging to the gut. And just as a human genetic connection, it was intriguing to me that out of all the GWAS studies that came out of inflammatory bowel disease, One particular loss of function variant that associated with IBD was actually the virus sensor MDA5, suggesting that a full functional MDA5 is actually important for protection in the gut. And now we can sequence viruses very easily using metagenomics. And that has led to a slew of papers showing that the so-called virome, the prokaryotic and eukaryotic viruses in our human tissues, are altered in a number of human diseases. And I've listed some here, but particularly in Crohn's and ulcerative colitis patients. But it was fair to say at that point, this metagenomic studies were vastly outpacing our understanding of really whether those changes in viruses contribute to disease, or really what is their immunomodulatory capacity in both healthy and disease states. And so we sort of think about how viruses are sensed, and usually this is in the context of an acute pathogenic infection. So many of us know that viruses are sensed by our innate immune system, either by cytosolic receptors, RIG-I and MDA5, or endosomal receptors, toll-like receptors, as well as many other helicases. And really the goal of this is to lead a signal transduction program that leads to the production of type 1 interferon and hundreds of interferon stimulatory genes whose role it is either to kill that cell or the virus directly. And so we asked, well, if this is a real antiviral defense mechanism, do those viruses that constantly exist in our tissues, such as the gut, do they trigger a similar or different pathway? And along those lines, many of the viruses in our gut are prokaryotic viruses that inhabit the actual bacteria. But there's more and more evidence emerging that actually those prokaryotic viruses can also have a transkingdom crossover and actually enter into our human cells, either by just simple uptake by phagocytosis, or uptake of the bacteria, and then the viral genome is there. Either way, there's evidence that these viruses can actually be sensed by our host innate immune system as well. And so, we sort of pose this very fundamental question of, is our immune system clever enough to distinguish between a virus that is going to hurt us or one that we've never seen before versus those that are happily existing amongst us? And so we asked, well, really in a sort of equilibrium when the virome that we are born with and sort of continuing to obtain over life, do we end up with an immunological equilibrium? And then what happens in disease? Do you get this change in virome that leads to a disruption in that immunological disequilibrium, or you actually get a very specific response to certain viruses that are contributing to disease? And so this was sort of a fundamental innate immune question that we embarked on with this project. So in order to do this, we struck up a fantastic collaboration with Rich Hoden at Mass General Hospital, who's a colorectal surgeon who unfortunately cuts out many, many colons from Crohn's disease and ulcerative colitis patients. 3/4 of Crohn's disease patients actually undergo surgery to remove that inflamed intestine. And so we obtained either fresh colon resections from Rich straight out of surgery, or actually obtained the fluid from the ileum of patients that had an ileum bag. And so the reason we wanted to focus at these sites, I think, was really twofold. We wanted to look really at the site of inflammation and look at the viruses there. But also, given that viruses are obligate pathogens and really do need host cells to replicate, we wondered whether there were different viruses actually in the intestinal tissue than one can detect in the feces. And so in order to isolate these viruses or enrich for these viruses, we did a process of size filtration. Now, this is by no means a perfect system. This is an enrichment for viral-like particles using a size selection. And then we did a chloroform, lysozyme, and DNase treatment to really get rid of any bacteria, any host DNA, and ended up with a fairly pure population of viral-like particles from these surgical resections. And so we could do electron microscopy with these and clearly see that they had morphology resembling both phage as well as eukaryotic viruses. And we could quantify these viruses by using cyber that would stay in DNA and RNA. And we also included dye for a phospholipid bilayer stain. And importantly for our immune experiments, we wanted to ensure that these viral-like particle preparations were endotoxin-free. So we did an additional step, and that was treat them with polymyxin B to get rid of any endotoxin. So the first thing we did was have a fantastic collaboration with Scott Handley at Washington University in St. Louis. So I would argue Scott is really leading the way in computational analysis of the virome. And so with Scott's help, we sequenced both the DNA and RNA from these viral-like particle preparations taken from non-IBD patient controls, ulcerative colitis patients, or Crohn's disease patients. And what Scott's team found was, in fact, as published in feces, many of the viruses in the gut were prokaryotic viruses, bacteriophage that were both double-stranded DNA, majority double-stranded DNA, or single-stranded DNA. But also in the tissue, we were able to detect a number of eukaryotic viruses from the families listed here. And I just want to draw your attention to this picornavirus family that we could readily detect in the intestinal tissue. And so once we started honing in on this picornavirus family, so this is a single-stranded RNA virus family, what was immediately obvious was actually all patients with IBD, either ulcerative colitis or Crohn's disease, had a significant elevation in enterovirus B viruses. Now, this was missed in previous fecal metagenomic studies, and so really sort of emphasized how the virome can be different depending on which area of the intestine you look at. And so here we mapped our reads back to annotated viruses, and we could see that the vast majority of these reads were coming from the echovirus 5 family, 100% identity, or this B75 family, or a lower identity to this E26 enterovirus B family. So as a reminder to you, picornaviruses are this single-stranded RNA positive sense virus, and they're about 7,500 base pairs in length, and generally is thought to be sensed by MDA5 in some preferential manner, although can be sensed by other sensors. The family of picornavirus is then split up into various genus, and then those species that we found were enterovirus B, and actually we did not see the Coxsackies, and the reason I bring that up is that Coxsackies have already been associated with type 1 diabetes and have a particular pancreatic tropism. What we saw was actually the echoviruses and enteroviruses that have tropism to the gut. And so we are very intrigued by this. And so is this a question of whether this particular virus is an environmental trigger or a contributing factor to the disease phenotype of IBD? And that's work undergoing. In addition to this eukaryotic virus, as published, we saw also changes in a number of prokaryotic viruses, bacteriophage, particularly in Crohn's disease, actually, over ulcerative colitis. And these are listed here. And interestingly, when we compared the virome in the actual colon tissue to what we found in the ileostomy fluid, it was clear that the richness of the virome was much higher in the actual tissue. Now, this is probably a combination of reasons. This is that viruses require host cells for their replication, so there's probably more viruses in the actual tissue. But also it's known that phage sit along the sort of mucosal layer of the intestine, and so we're probably isolating those as well. But you can clearly see that in the actual ileostomy fluid, which is even further up the GI tract than feces, obviously, the richness was reduced. But nonetheless, we still did see disease-associated changes, Crohn's disease here in red and ulcerative colitis here in yellow, some changes in the virome of the ileostomy fluid in these IBD individuals. And so Scott's team did a similar analysis on the ileostomy fluid virome. And here we did not detect the picornaviruses, but rather this was predominantly this single-stranded DNA anellovirus. And there was some inkling that maybe some of these actually are reduced in IBD, which is an interesting idea that some of these protective viruses actually are reduced in disease. And we saw as well a number of phage in the ileostomy fluid. One phage that we actually saw that was specifically enhanced in patients without IBD was a phage that is predicted to occupy Akkermansia. And Akkermansia is already known to be a protective bacteria in the gut, so we found that quite interesting that this particular one was elevated. But we also saw some go up in IBD, and those are listed here, usually from the Microviridae phage family that were elevated. So now we've identified these viruses. The challenge is really in this field is being able to culture individual viruses to work with, and that applies to both the prokaryotic viruses and the eukaryotic viruses. Many of those that we can identify by sequencing, you can't actually work with them because many of them aren't culturable yet. So we took the approach of just wanting to ask, well, what is the overall immune response to the collective viruses that we isolated in these human colon resections? So to do this, we took primary human macrophages from healthy donors, just healthy blood donors, and then we put either the non-IBD viruses, the ulcerative colitis viruses, or the collective Crohn's disease viruses on these primary human macrophages. And the first thing we could see was that these viruses were certainly taken up into these macrophages quite readily. So if we labeled these viruses with cyto16, we could clearly see that they were taken up. And this was dependent on phagocytosis, because if we did a Golgi block such as Profeldin A, we didn't see this uptake. And so the first thing to note is that the uptake between the non-IBD, UC, and CD were equal. So anything I'm about to show you was not because one had more uptake than the other. And so we took the macrophages after delivering these viral-like particles from the intestinal resections to them, and then we performed some sequencing. And so the RNA-seq really was quite an initial profound result. So that is looking at the MDS plot of the transcriptional program that was induced in macrophages for non-IBD individuals. So these are 5 individual patients. Really, the transcriptional program induced by those viruses was quite consistent, but was markedly divergent from that transcriptional program that was induced by either ulcerative colitis or Crohn's disease viruses that actually very well, they overlapped quite well in the transcriptional response that they initiated. It's also worth noting that these non-IBD viruses from a healthy, uninflamed gut actually were not silent. They actually did induce something different to a mock control. So, but it was just very divergent from the IBD viruses. And if we looked at that in another way, we could see that the healthy virome upregulated a number of genes, that very few of those overlapped with what was upregulated with the IBD virome. What was interesting is that the healthy virome seemed to have this downregulation of the transcriptional response in macrophages, suggesting that it was kind of dampening the host immune response. And this had very little overlap with the ulcerative colitis or Crohn's disease viromes that really did overlap quite tightly in the transcriptional programs that they induced. So what were the genes induced in macrophages by these respective viromes? As you can see, that the healthy virome very much dampened the interferon response. So these viruses collectively downregulated a number of interferon stimulatory genes, a number of toll-like receptors, a number of IFITs, etc., were all significantly downregulated in macrophages by these viruses. And what they did upregulate was actually a very atypical program. So this was certainly not a type 1 interferon program. It was more a sort of pro-survival, resolving macrophage type program. So things like CISH, IL-4 receptor, pro-survival factors such as BCL2, the Wnt pathway, etc. And this was analysed by gene set enrichment analysis, where you could really see this down-regulation of all these interferon pathways and actually an upregulation of these pro-survival pathways. In contrast, the ulcerative colitis viruses on macrophages very much provoked inflammation. So all the classic cytokines and chemokines that one would expect from an innate immune response, things such as IL-6, IL-8, interferon gamma, even IL-1 alpha, IL-1 beta, chemokines, etc. And these were all fit into these pro-inflammatory pathways. And Crohn's disease viromes were very similar. So these collective viruses, again, triggered this very much this pro-inflammatory pathway in macrophages. So I think this was the first evidence that taking the viruses from IBD patients where we know that virome is altered, you put it on an innate immune cell and those viruses very much provoke inflammation. So it suggests that these viruses are certainly contributing to the inflammatory environment of inflammatory bowel disease. If we started taking the view that they may polarize macrophages in different ways, we started to look at classic so-called M1, M2 genes. And you can see here in blue, the healthy viruses very much downregulated sort of the M1 genes and upregulated these M2 polarizing genes, whereas the ulcerative colitis and Crohn's disease viromes pretty much provoked a sort of M1 pro-inflammatory polarization. And we confirmed many of these with many more viruses from many more patients. I think we got up to 14 patients per group. These were all colon resections taken fresh, the viruses isolated from, put on macrophages and cytokines measured. And what was actually amazing was that the reproducibility of the virome from individuals. So you can see here the amount of this anti-inflammatory cytokine interleukin-10 was quite consistently upregulated by non-IBD viromes. The anti-inflammatory interleukin-22, again, very much upregulated by non-IBD viromes, as well as this pro-survival gene CISH. And importantly to us, we wanted to know whether this was direct sensing of the nucleic acid of these viruses we isolated. And so we took the VLPs, we isolated RNA or DNA, and we transfected that into the macrophages at equal concentration. And even when we put equal concentration of the RNA or DNA, you could very much see that the healthy viromes triggered this anti-inflammatory interleukin-10. In stark contrast, the IBD viromes very much provoked inflammation, very reproducibly upregulating TNF, IL-6, as well as more interferon beta. And importantly to us, from innate immunology perspective, we wanted to really ensure that none of this had to do with any contaminating endotoxin. And so we repeated the same experiments in toll-like receptor 4 knockouts. So we took bone marrow-derived macrophages from toll-like receptor 4 knockouts, and we could see that the amount of interleukin-6 triggered by Crohn's disease or ulcerative colitis viromes was equal in the absence of toll-like receptor 4, suggesting that this was completely independent of this pathway. And then similarly, we isolated RNA and DNA and transfected that in as equal amounts, and we could see that the IBD viromes very much provoke TNF production, even when transfected in at equal amounts. So if you think about the innate immune system, what is sensing healthy in IBD enteric viromes? Well, we know quite a bit about how the immune system sees viruses, either just double-stranded or long double-stranded RNA in the case of MDA5, single-stranded RNA, perhaps through NOD2. We know for RIG-I, it's definitely this triphosphate sort of panhandle RNA that this particular viral sensor recognizes. And of course, for DNA, either through CGAS, TLR9, etc. And so we just wanted to know, well, what was the viral moieties that were detected by the host immune system? And the first thing was that we knew that these responses we saw in macrophages were dependent on innate immune sensing, because if we took knockout cells for MABs, which is the downstream adapter for MDA5 or RIG-I, we had a reduced response. If we knocked out CGAS, we saw a reduced response. If we knocked out MyD88 downstream of toll-like receptors, we lost the response. But again, this response was intact in toll-like receptor 4 deficient cells. And so all of this immunomodulation we're seeing is dependent on innate immune sensing, either by cytosolic or endosomal toll-like receptors. If we treated the RNA from these viruses with RNAs or calf intestinal phosphatase to get rid of phosphate groups, we lost that response. So we for sure, it's sort of known RNA, DNA moieties that are being sensed by the innate immune system of these viromes. And again, we knew CGAS was involved if you transfect DNA in, because we lost that response in the absence of CGAS. And so this was an interesting observation for us, that the first time that the virome from a healthy gut was actually protective, could trigger this anti-inflammatory pathway, whereas the IBD virome very much provoked inflammation. So we sort of asked the question, well, if that's the case, maybe in a similar way to fecal transfers of microbiome, the virome or the healthy virome could be used as a therapy. So could we suppress the inflammatory response induced by IBD viromes by delivering healthy viromes in increased concentrations? And this is just in vitro, but in human macrophages, in red, we could see that the IBD virome triggered substantial interleukin-6. And as we increased the amount of healthy viruses on top of that, the healthy viruses not only on their own could trigger anti-inflammatory responses, but actually could start to suppress the inflammatory response induced by IBD viromes. And we could see this as well with TNF. And we asked whether this was a direct effect of the viruses, or this was because of anti-inflammatory mediators that we'd already seen were induced. So we took the supernatant from those macrophages previously infected with healthy viromes from patient colon resections, and essentially asked whether that supernatant could also suppress the response to an IBD virome. And so in light blue here, you could see that definitely whatever is in that supernatant, likely interleukin-10, interleukin-22, could readily reduce the inflammatory response driven by the IBD virome. If we heat inactivated that, that would get rid of soluble proteins. We lost that effect. But if we UV inactivate that, which would cross-link viruses, we could still see an effect. So it's very much suggesting that it's anti-inflammatory mediators in that supernatant, driven by the non-IBD virome that could be protective. So this sort of suggests to us that much like the microbiome, perhaps virome transfers could be considered for IBD therapy, where healthy viruses delivered could actually be suppressive of inflammatory responses. And it's worth commenting here that in C. difficile infection, this has actually already been shown, where fecal transfers or fecal contents were filtered out for bacteria, and the filtered product was delivered, and actually that also showed to have an effect on C. difficile infection. So the non-bacteria component of fecal transfers has already been shown to be able to immunomodulate and be protective. So all of what I've shown you is in cells. We did this also in intestinal epithelial cells to show this divergent effect. But we really wanted to know in vivo what these viruses could do. So we humanized mice with viruses that we'd isolated from colon resections from non-IBD patients, ulcerative colitis or Crohn's disease patients. And to do that, we first treated these mice with an antiviral cocktail, which is a published cocktail to sort of reduce the murine virome. And that did about a 20-fold reduction, or we saw about a 20-fold reduction in the viruses present in an SPF Black 6 mouse. Then we delivered by gavage these human viruses that we'd isolated from these colon resections, and we gave that by 3 gavages. And we could quantify that here, either in the colon tissue itself or in the feces. The antiviral cocktail got rid of viruses, and we got actually quite a decent amount of viruses in the feces as well as the colon tissue following this gavage. So the first thing we asked was there any spontaneous ability of these viruses to affect the host immune system? And so we took lamina propria immune cells after the gavage and just looked whether these viruses were capable of changing immune cell populations in the lamina propria of these mice. And so if we looked at CX3CR1 positive mononuclear phagocytes in the gut, which are a known pro-inflammatory phagocyte in the gut, we very much saw that the IBD virome expanded this population spontaneously when we delivered these either ulcerative colitis or Crohn's disease viromes to these mice. And we could quantitate that both by percentage as well as total counts. We saw this significant expansion of CX3CR1 positive mononuclear phagocytes by delivering IBD virome to these normally healthy mice. In contrast, what we saw was actually the non-IBD virome in blue could actually enhance the numbers and percentages of these CD103 positive mononuclear phagocytes, which have been shown to actually be able to promote T regulatory cell expansion in the gut. So on the one hand, the IBD viruses spontaneously expanded inflammatory monocytes, whereas the healthy virome, it led to an expansion of these more protective CD103 positive mononuclear phagocytes. And so then we wanted to induce colitis in these mice and ask, was the healthy virome able to protect these mice from colitis? And in contrast, was the ulcerative colitis virome or Crohn's disease virome capable of exacerbating chemical-induced colitis in these mice? And so one crude readout for intestinal inflammation is just simply measuring colon length. So the shorter the colon, the more inflammation. And so in grey was the mice that had been treated with that antiviral cocktail. And we sequenced those mice, and we definitely saw that there were alterations in both the virome and the microbiome in those mice. But as published, that antiviral cocktail really does exacerbate intestinal inflammation. So taking away viruses from mice has repercussions for intestinal health. What we did see, though, was the ability to rescue that quite beautifully by gavaging in those healthy viruses, the viruses taken from a healthy intestine. We could see that they very much protected these mice from induced DSS colitis, whereas the IBD viruses could not do that. Another readout of intestinal inflammation is looking at lipocalin in the stool, which is usually a marker of neutrophil infiltration. Again, taking away viruses with the antiviral cocktail increased stool lipocalin significantly, and we could rescue that quite beautifully by gavaging those healthy viruses into that mice, whereas the IBD viruses could not rescue the stool lipocalin. Similarly, if you measure the pro-inflammatory cytokine interleukin-6 in the actual colon tissue, we could see that the taking away the viruses increased interleukin-6, putting healthy virome back into these mice, rescued that, and that could not be rescued by the IBD viruses. And a number of other readouts, we looked at histology, which was blindly scored by a pathologist, very much reduced inflammation here in blue in the healthy virome humanized mice, whereas the IBD viruses, virome mice very much had inflammation. Interestingly, too, in addition to just inflammatory cytokine production in the gut and infiltration of leukocytes, we looked at intestinal leakiness of these mice. So you can gavage in fixie-dextran, and if the gut is leaky, you can then measure that in the blood. And actually, in yellow and red here, the IBD virome very much provoked sort of intestinal leakiness, which is another sort of indicator of inflammatory bowel disease. So collectively in vivo, these human viromes contributed to disease state. So on the one hand, the healthy virome protected the gut in vivo, and on the other hand, either ulcerative colitis or Crohn's disease viromes very much provoked inflammation in vivo. So suggesting that the changes with metagenomic studies seen in IBD, these viruses are actually contributing to disease phenotype. So the next question we asked was any of this dependent on host sensing of the nucleic acids of these viruses? So in other words, in vivo, of course, we can't exclude the contribution of the microbiome by delivering these viruses to these mice. And we did certainly look at 16S sequencing. We did see some changes in the microbiome, but actually only really for the Crohn's disease delivered viruses. So we went back to our mice and we wanted to ask specifically, with all of the work we've done in human macrophages showing the divergent innate immune response to these viruses, was this relevant to the different disease phenotypes that we'd seen in vivo? And so we took either MABs knockouts that cannot signal downstream of RIG-I or MDA5 viral sensors, or CGAS knockouts which cannot sense DNA. And in both cases, MABs knockouts or CGAS knockouts, we really lost any immunomodulation of these human viruses that we were delivering via gavage to these mice. So we didn't see really any effect of these human viromes that we had delivered to the mice in the absence of either RNA sensing or DNA sensing. So this is really strong evidence that the innate immune system is really the key sensor of these viruses, both in health and disease. So on the one hand, for any benefit of the healthy virome, you need sensing by the innate immune system, or any inflammation triggered by the IBD virome, you need sensing by the host innate immune system. So that was all in mice, and we'd done human macrophages in vitro, and we'd done looked at humanized mice in vivo to look at disease phenotype. And I sort of wanted to get back to this genetic association of the viral sensor MDA5 with inflammatory bowel disease. So many of you on this call and many of you in this field know that MDA5 is a cytosolic sensor of long double-stranded RNA. And in many autoimmune diseases, actually so-called interferonopathies, you see a gain of function actually of MDA5 that contributes to these diseases. And in IBD, actually what we see is the opposite. It is a loss of function variant of MDA5 that uniquely associates with inflammatory bowel disease. So this suggested to us that this, shows us that MDA5 in the gut, at least, is really needed for maintaining intestinal health in response to the virome. And so in pink here are the main mutations that associate with inflammatory bowel disease. So it was either this isoleucine 923 is in the C-terminal domain is mutated to a valine, or you have a splice variant in the C-terminal domain, or you have this truncated mutant, which essentially truncates here. So all of these end up with a lack of function C-terminal domain. And we wanted to confirm that these are truly loss of function. And so we just expressed these mutants in HEX cells and found that indeed these are loss of function mutations. And so just to take you through this, either expressing wild type MDA5 to no RNA or self-RNA, where we'd isolated RNA from the cytosol of the same cells, triggered or did it on its own without RNA, triggered an interferon response. But in the presence of a known MDA5 ligand poly-IC, that interferon response was exacerbated. And we also isolated our human VLPs from colon resections and showed that in the same luciferase assay, they could trigger an interferon response that was MDA5 dependent. But the mutants, either the truncated version or the I923V mutation, all inhibited the ability of these healthy viruses or poly-IC to induce a sort of a downstream response of MDA5. And so with John Ryu up at Montreal, we then took a step further where we isolated iPSCs from patients that were carrying these MDA5 loss of function mutations. And John's group has a beautiful system where they can take those iPSCs and convert them to essentially spheroids or organoids, and then we can create either these organoid cultures or these 2D cultures. And we did this with intestinal epithelial cells because MDA5 actually is predominantly expressed in the intestinal epithelium. And so what we saw was an ulcerative colitis patient that had this compound, was a compound heterozygote actually for both of these mutations, in a loss of function of MDA5. And we could see that under normal circumstances, that intestinal epithelium looked fairly normal. With E-cadherin or Z01 or F-actin, that mutation really didn't change the intestinal epithelium in any obvious way. But in a 2D culture, when we started delivering either healthy virome or IBD virome, we could start to see this very significant loss of epithelial integrity barrier in the sort of presence of this MDA5 loss of function mutation. So without MDA5 functionality, the virome was much more damaging. And even if you put an IBD virome on top of that, you get that additive effect where you've sort of got a genetic susceptibility gene plus an IBD virome results in a lack of intestinal barrier integrity. And we looked at the ability of these intestinal epithelial cells to produce the protective cytokine interferon lambda. And we could see that the healthy virome triggered interferon lambda production And this was lost in that patient that was carrying this compound heterozygote mutation within MDA5. So this is some human evidence that MDA5 is necessary for sensing of the virome, and actually loss of function variants in IBD patients actually lose any beneficial effect of the virome, but also when you have an altered virome on top of that, it leads to even more exacerbated inflammation. So in summary, I think we've done a really large foray into trying to understand the human intestinal virome. I think up to this point, really the virome was lagging very behind the microbiome, largely because of the techniques in which we detect it. And so once sequencing became cheap and easy and ways to computationally align viruses, much data has come out of associating viruses or changes in the virome with disease. But we were still really left with correlative data. The question we took or asked was, what does the virome do to the innate immune system in health? And what does it do in the context of inflammatory bowel disease? So I think our data has really clearly shown in both the reductionist system, where we just put the viruses we isolated from the intestinal colon on human macrophages in a dish, the healthy viruses very much provoked an anti-inflammatory pathway, which was an atypical viral response. So it wasn't a type 1 interferon-induced response. Actually, that, if anything, that was down-regulated, and instead what was upregulated was this pro-survival anti-inflammatory pathway. And in stark contrast, the IBD viruses very much provoked inflammation. So I think we can confidently say from the human macrophage experiments that a reductionist system, viruses on their own can actually trigger inflammation. So the IBD viromes trigger inflammation. And then even in vivo, when we humanize those mice, very much the healthy virome was protective, whereas the IBD virome provoked inflammation and actually even spontaneously provoked that expansion of those CX3CR1 positive mononuclear phagocytes. So I think we're at a juncture here in virome research. I think we've now shown that the immune system certainly doesn't ignore these viruses in health, and these viruses that are altered can contribute to disease phenotype. The questions we have moving forward is really, is it that it's the change in the virome? Specifically, is it certain appearance of viruses such as the enterovirus B family that's really triggering this inflammation? That's a very, very likely hypothesis or outcome. But the same or sort of a separate hypothesis is really that whether the same viruses could be doing different things depending on the immune state. And we've seen this for certain bacteria where a so-called symbiont can become a pathobiont in the context of inflammation. And so we're very interested in this concept of whether the IBD virome triggers inflammation because of the appearance of enteroviruses and other phage that aren't normally seen in healthy state. But at the same breath, do some normally healthy viruses, such as anelloviruses, say, actually start being pro-inflammatory in a pro-inflammatory environment? And these are the types of questions that we'll be asking moving forward. I think what was really convincing for me was this genetic association of the virus receptor MDA5 with IBD. So we showed quite clearly that that is a loss of function mutation in MDA5 that associates with IBD. If we took intestinal epithelial cells from a patient that it was a compound heterozygote for that loss of function, clearly there was detrimental consequences for that. So you really do need a functional MDA5 receptor sensing the healthy virome, and really when you lose that, you can actually, there's detrimental consequences for the intestinal epithelial cells. So I think collectively our work really does show that the virome is not ignored. The healthy virome certainly is not ignored, and in a similar way to the microbiome actually works to educate our immune system and keep sort of our intestine in a healthy state by provoking sort of these anti-inflammatory pro-survival pathways. And it can do that autonomously to the microbiome. And in contrast, the IBD virome very much provokes inflammation. And so the idea here is, well, can we take this a step forward? On the one hand, can we start to harness the healthy virome in a similar way to the microbiome to sort of suppress inflammation in IBD? But then on the flip side, what can we do about the IBD viruses? And so can we start to try and eliminate some of these viruses in IBD that we think are contributing to this inflammatory phenotype? And I think this will be really the next frontier in virome research. And so with that, obviously, I get to tell all of you about the work we've done. This has been the hard work of many people in my lab, but particularly Fatima Adili-Agdam, who was just a stellar postdoc who really led this project. Fatima is now training to be a surgeon herself. So after all this work looking at colon resection, she's now actually getting to do some surgery herself. So I look forward to having her as a physician surgery scientist colleague in the future. I want to point out Hajra Amatullah, another postdoc in the lab, as well as Isabella, and really everyone in the Jeffrey lab that helped us with this project. This was a new frontier for my lab, so really collaborations were key. I want to point out particularly Scott Handley, who really, again, I just want to emphasize, is really leading the field in virome computational analysis, and we couldn't have identified all those viruses in those surgery resections without him. I also want to give a shout out to Ramnik Xavier and Rich Hoden. Ramnik Xavier at MGH has set up an enormous registry of IBD patients at Mass General Hospital, so it allows us researchers access to tissues, blood, whatever we like, biopsies from these genotyped individuals, which has been just such a fantastic resource to try and understand inflammatory bowel disease. And of course, computational help, as well as John Ryu's team in Montreal, who made the intestinal epithelial organoids from these MDA5 loss of function patients. So I'm obviously not there live. Thank you everyone for your attention. What I will say is that I'm very happy to be answering questions via email. So if you have questions about this work and the field more generally, please reach out and I'll respond as soon as I can. And thank you for your attention.
I want to thank you, Dr. Jeffrey, for your time today and for your important research. And before we go, I want to thank our audience for joining us today as well. Questions submitted today and during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until September 7th, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.